Hello, everybody. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Jennyfer Mora Mitchell. I'm a postdoc at the University of Colorado Anschutz Medical Campus, and I will be moderating today with Dr. Sonpho, a postdoc at Boston Children's Hospital, Harvard Medical School. We're excited to highlight the work of our standing postdoctoral members. Today, Dr. Ekasit Sonpho from Stowers Institute for Medical Research and Dr. Katharine Goodwin from MRC Laboratory of Molecular Biology will share the research. Each speaker will have 20 minutes talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box and we will read them for you. I am pleased to introduce Dr. Ekasit Sonpho, a current postdoctoral researcher at the Stowers Institute for Medical Research in Dr. Alejandro Sanchez Alvarado's laboratory. Ekasit's academic journey began with his Bachelor's of Science in Biology, which he pursued under the distinction program at Mahidol University in Thailand, where he also earned his PhD. Dr. Ekasit has co-authored a Nature Communications paper and is the first author of 2 exciting papers in ECM research published in the journals Zoological Science and Molecular and Cellular Proteomics. Since 2016, Ekasit has shared his research through multiple oral and poster presentations. His work with planaria, from studying drug effects on behavior and regeneration to creating planarian bodies from isolated neoblasts by FACS, reflects his unwavering dedication. Since 2011, Ekasit has received multiple travel awards and scholarships from different programs and universities, like the Japan East Asia Network for Exchange of Students, Mahidol University, and the Osaka University Student Services Organization. This last award allowed him to do a 5-month exchange program at the Frontier Lab at Osaka with Professor Tomoji Mashimo. He also obtained 2 awards to work with Professor Agata Kiyokazu. In 2018, Dr. Sonpho received the Fulbright Junior Research Scholarship to visit the Alvarado Sanchez Lab for the first time. He also was one of the 10 finalists in the FAME Lab Thailand, a program equivalent to the 3MT competition. More recently, Dr. Sonpho received a travel award from the American Society for Matrix Biology to present his research at the 23 meeting. Dr. Sonpho returned to the Sanchez Alvarado Lab in 2021 to continue research in planaria and extracellular matrix biology, which he will present today under the title Multiple Cell Types Influence Extracellular Matrix Dynamics During Planarian Regeneration. Dr. Sonpho, please take it away.
Thank you for a nice introduction. So let me share my presentation. Okay, so thank you everyone for being here, and I am really excited to present the data that we have so far. Let's begin with the tales of Greek mythology, which is the epic fight between the Hercules and the water serpent, so-called Hydra. The Hercules was assigned by the king to kill the Hydra, but whenever he tried to amputate the Hydra head, Humor just regenerate back. This myth is a good example of how humans is so obsessed with regeneration process. And I'm not blaming them because we observe many animals regenerate and survive from the deadly injuries. Regeneration is a constant process that involves with replacing, repair. And it has been conserved throughout the animal kingdom. But what's special about regeneration is there is one common denominator, which is the stem cells. The stem cell is the versatile cells that are able to differentiate into various cell types and fuel the regeneration process. Nevertheless, the knowledge of it, however, it needs to be acknowledged that stem cell is not working alone. It needs to pair up with some type of cell. In this case, is a fibroblast. Fibroblasts will provide a suitable microenvironment and fuel the healthy tissue regeneration. The fibroblast is a part of connective tissue that was studied and found to be involved in homeostasis of the tissue, regeneration, growth, patterning, and even the disease progression, such as a fibrosis. Nevertheless, the knowledge we have so far about the ECM biology are long depend on the vertebrate model, such as mouth and zebrafish. Although this vertebrate model provide a fruitful information, it still has a lot of limitation. For example, the tissue is really complex and sometimes regeneration is really restricted. In order to get more profound information about how ECM regulates the stem cell, I would like to embrace everyone to the invertebrate model that accounted just only 2% of the ECM research so far. These surprising numbers makes me curious. How come there are so little research that focus in ECM in invertebrate? Is that because invertebrate lack ECM? I don't think so. For my quick search, invertebrate has ECM, at least based on the literatures such as the microscopic investigation or the cell culture, which is quite a shame considering the fascinating and awe-inspiration biology that invertebrate has to offer. Here are the examples. The nematostella has abilities to regenerate the whole body. The sea star has abilities to regenerate the losing arm. And like this cute apple snail has abilities to regenerate the whole camera-type eyes. But for today's talk, I would like to shift your attention to this model, Smithia mediterraneans, or the flatworm, the planarians. Planarian has remarkable abilities to regenerate. Imagine that you will cut a piece of the tails of this worm. This piece of tail regenerate into fully grown adult within 2 weeks. For those who never seen a planarian before, don't be worried. Imagine that you have the dime on top of your hand. And imagine that this is the worm, the size related to the dime. The worm is small, but is still visible with your naked eyes. The secret behind these remarkable abilities is the body is filled with stem cells, accounted for 15% of the cells. And these stem cells are able to differentiate into many cell types and make regeneration possible. With such a large amount of stem cells inside the body, you can imagine that how tight control the microenvironment need to be in order to keep everything in place. Otherwise, instead of having the cells that divide in the orderly fashion, you will end up with something like a giant tumor in the worm, and the worm hates that. So you might ask, how many percent of the cell that accounted for the fibroblast that has a function to maintain the microenvironment? Surprisingly, the answer is none. They do not have a distinct type of a fibroblast. This observation is the fundamental question that I am interested in. If not a fibroblast, what kind of cell that have a function to maintain stem cell microenvironment? Although the morphology of the planarian is flat and simple, the tissue architecture is impressive. They have many cell types that form many complex organs. In 2019, scientists suggest that the muscle act as the connective tissue based on the fact that the muscle secrete a lot of collagen, which will compose the core collagen component in the animal tissue. Nevertheless, we also observe that there are non-muscle cell types that are able to secrete the ECM as well, such as epidermis, intestine, even a neuron. So this is my working hypothesis. I hypothesize that several cell types need to work collaboratively to create a favorable niche for stem cell to flourish and participate in tissue regeneration. To start my project, we need to verify that the ECM indeed expresses the ECM transcript. So I conduct in situ hybridization to locate mRNA of the ECM genes. We found that many ECM genes is actually transcribed in the planarian body. In order to confirm this finding, I performed double in situ hybridization. The magenta probe will represent the intestinal marker, and the yellow probe will represent the ECM transcript. We find a strong colocalization between ECM and the intestinal marker in the intact animal and the animal that undergo regeneration. Unfortunately, the mRNA expression does not always reflect the protein expression. There are many post-transcriptional and post-translational modifications that would activate or inhibit the protein activities. And especially we're talking about the ECM, it needs to be heavily modified and transport outside the cell, make another layer of the complexities. To better understanding the dynamic of the ECM in many levels, I combine two separate omics, the transcriptomics and proteomics. For the transcriptomics, the RNA from regenerating tails animal bone extract and submit for the RNA sequencing. For the proteomics, the ECM from regenerating animal will isolate by the technique that we call tissue decellularization. This technique allows you to extract the cells and leave the protein scaffold behind. By the end of the experiment, we are able to compare the mRNA expression and ECM protein expression during planarian regeneration. But one of the challenges in proteomics is that the amount of the protein need to be high. Essentially, the more protein, the better. To maximize the amount of ECM that we use in the experiment, because the worm is so tiny, we developed the decellularization chamber that allow us to isolate up to 400 animals per day. And the result is what we kind of expect. There is a big conflict between the transcriptomic expression and the proteomics expression. Some protein express the mRNA in the early stage, and the protein just somehow following 2 or 3 days after that. To get more clear pictures of the dynamic is, we classify the candidate into 2 groups. The magenta group will represent the candidate that has the protein peak at the late stage of 5 to 2 weeks post-amputation. In the other hand, the yellow box will indicate the candidate that has a protein that peak at the early stage or any day before 3 days post-amputation. So we're wondering what kind of cell type that required to build the ECM at the different phases. Therefore, we try to infer the cell identities by using our pre-existed single-cell RNA sequencing. We found that more than half of the ECM that produced at the early stage were derived from epidermis, intestinals, and even the stem cell itself. In the meanwhile, it seems like the muscle is responsible for ECM production at the late phase. To imply and investigate any potential function of the early phase and late phase ECM, we utilize gene ontology enrichment analysis. We found that the ECM that express at the early phase are enriched for the protease activities. In the meanwhile, the ECM that produce at the late stage are enriched for the structural organization function. And this aligns with our expectation because we are expect to see the protease activities at the early phase of regeneration, that including the wound healing process or clearing the tissue or clearing all tissue architectures and provide the open space for the tissue to build up the new tissue architectures in the late phase. Now, we learned that the non-muscle cell can actually secrete the ECM, but one of the most important questions is, do they actually function and impact the stem cells? We decided to use sublethal irradiation assay where the low dose of gamma irradiation were used. The gamma irradiation will wipe out any dividing cells, but with the low dose, it will leave some population behind. And if we will let those survived cells to repopulate and see how fast the stem cell repopulate. To study the function of the genes, the only way we can do that is use double-strand RNA feeding, or we call RNAi feeding experiment. We fed them with the RNAi for 6 times. We do a sublethal irradiation, and we collect the sample of the worm 10 days after that. The readout would be the density of the stem cell after 10-day post-irradiation. We know that UNC22 is the negative control RNAi. This is a gene that does not exist in planarian genome, so we use it as the negative control. So this is the graph that represents the fold change of the stem cell densities relative to the control, which display at your most left-hand side. The result is really clear. It suggests that some of the ECM, once knocked down, has both positive impact and negative impact to the stem cell repopulation after the sublethal irradiation. And this is reflected by how fast the stem cell repopulate after the sublethal irradiation. We keep to continue to monitor our RNAi feeding animal, and we found that some of them never fully able to recover. They develop the tissue lesion, head regression, and finally they die. And during the process of the screening, we also identify one unknown protein that seems to be specific to planarians. And this protein is under our radar, and we are under the characterization process. The second assay that we use is tissue regeneration assay, where we amputate the animal after 2 rounds of RNAi feeding, so that in total we do is 12 times of feeding. The readout would be the stem cell proliferation by using phosphorylate histone H3. And this marker will mark the cell that undergo cell division process. The result is similar with the previous assay in that many ECM components are important for stem cell proliferation after tissue injuries. The outcome of this assay, we found that we identified the glutathione peroxidase is the stem cell regulator that once knocked down, the blastema cannot fully regenerate, and it is undergoing the characterization. In conclusion, we currently propose the model that planarian exhibit remarkable dynamics between the mRNA and protein level. Many cell types is involved in such process. Intestinal and epidermis are likely to involve in the early phase of regeneration by degrade the protein, while the muscle will act as the late phase regeneration and produce the core collagen for build up the new architectures. This project might reveal the insight of how the early metazoa can evolve that has not evolved a special cell type to produce the ECM, maintain a suitable environment for regulating stem cell activities. However, speaking of the fibroblast, I would like to share our thought about the definition of a fibroblast that used by our communities. Originally, fibroblast has been discovered by the pig embryo, and the term fibroblast has been coined after that discovery. However, when we trace our way backward throughout the phylogenetic tree, the definition of fibroblast becomes more ambiguous. In case of planarian, I have shown you the great example of the muscle that act as the connective tissue. So should we call the muscle as a fibroblast, or should we just say fibroblast is not exist in planarians? And I would say this is one of the debate that we are currently have in our own lab. And now let's talking about the mammalian fibroblasts. The mammalian fibroblasts are more distinct. They have much more well-defined gene marker. For example, the pellet-derived growth factor alpha. But for this marker in mammal, this genes is not clearly to be exist in planarian. So how should we define the cell that actually responsible for ECM secretion in the organism? Since most organisms in the tree of life seem to be able to secrete the ECM at some point. Therefore, I would like to propose and I would like to redefine how we call the cells that produce the ECM as the metrogenics. This translates directly into the cells that produce the ECM. However, whether the metrogenics is just the new cell state or it's just the transient cell or it's just a transient cell state, or it is an entirely new cell type, it requires further investigation. And by that, I would like to end my presentation. I would like to acknowledge everyone in Alejandro lab, and especially Alejandro himself. He is a very supportive mentor that open for new possibilities and new experimental design. And all of these experiments will not be possible without the help from the personnel from the technology center, including system mass spectrometry, microscopy, invertebrate department who take care of our worm really nicely and fondly, sequencing team, media prep. And by that, I would like to take the question from the audience, and thanks for your attention. Thank you.
Thank you, Dr. Sonpho. So while we're waiting to get some questions in the Zoom box, I would like to ask you first, the fact that we don't see ECM in placozoan, does it mean that there is also no fibroblast cells in placozoans?
Can you repeat the question again?
Yes, the fact that placozoan doesn't have an ECM, seems like it's the only group that doesn't have an extracellular matrix. Does it mean that they also don't have fibroblasts?
I would like to, I want to admit that I cannot find the evidence that suggests that the placozoa has an ECM. They may have the ECM, I'm not sure, and I definitely not sure about the fibroblasts. But if anyone has any idea if the placozoa has an ECM, please just let me know, because I really would love to update my phylogenetic tree.
Okay. And there is another question coming in the box, so it reads, let me see. How do you think about linking protein level to mRNA level and then extrapolating that to previous single-cell RNA sequencing data? The protein measurements are on both samples, so do you have thoughts about possible different kinetics in different cell types?
Yes, to answer about the bulk proteomics data, I did not do that, but that is actually a good suggestion. And what is the second part of the question again?
Yeah, let me read it to you. The protein measurements are on bulk samples, so do you have thoughts about possible different kinetics in different cell types?
Yes, we have. The reason that we need to infer the cell identity based on the RNA sequencing is the single cell proteomic is not yet available, but I have heard that the scientists in the mass spectrometry team is already get into it and try to make that technology possible in our institute. So I really would love to study the cell identity and cell dynamics in each cell type.
Right. There is another question. Muscle cells in planarians also secrete lots of morphogenes for polarity, like Wnt, BMPs, and alike. Have you looked at whether muscle ECM helps those signals to be sent and received properly? And thank you, and this was a fun talk.
So for the muscle in planarian, it has the function to control the polarity of the worm as well. It is not my direct interest, but it has been experiment that knocking down the genes that secrete by the ECM, such as Wnt1 or another morphogen, actually impair the abilities of the worm to the polar axis determination.
Great. I don't think I see more questions coming, so I think we are good to continue with our next speaker.
All right. Well, thank you, Jennifer. It is my great pleasure to introduce the second speaker for the seminar today. My name is Wade Sugden. I'm from Boston Children's Hospital, and our second speaker is Dr. Katharine Goodwin. Katharine has had a longstanding interest in developmental biology, biophysics, and computational biology, and has pursued questions in these areas in several model systems over her career. She did her undergraduate studies in the quantitative morphogenesis lab of Rodrigo Fernandez Gonzalez at the University of Toronto, where she studied three-dimensional cell morphology changes during axis elongation in Drosophila. For her master's at the University of British Columbia, she worked on extracellular matrix adhesion and developmental tissue mechanics in the lab of Guy Tannensauf. She joined the lab of Celeste Nelson at Princeton University in the United States for her PhD in 2017, where she studied diverse aspects of lung development in the mouse, including airway epithelium morphogenesis and smooth muscle differentiation. In this work, she showed that smooth muscle wrapping is required to shape airway epithelium into branch networks, and that smooth muscle cell plasticity exists in mouse developing lungs, which can be uncovered by analysis on a background of deficiency for the transcription factor SRF. Since 2022, she's been doing her postdoc in the lab of Kate McDowell at the MRC Laboratory of Molecular Biology in Cambridge, UK. Here, she's been applying adaptive light sheet microscopy to study early post-implantation development in the mouse embryo, including the migration of primordial germ cells, which will be the focus of her talk today. Katharine has authored several first author publications in high-impact journals like Development and Developmental Cell, and received numerous awards and independent funding, including the American Heart Association Pre-Doctoral Fellowship and the Princeton University Proctor Fellowship. She has also taken an active role in mentoring undergraduates, teaching and outreach to high school and primary school students to speak about microscopy and developmental biology. I could say more, but with that, I'll stop there. Thank you so much for being here, Katharine, and we're looking forward to your talk.
Thank you so much for that kind introduction. I'm just going to share my screen real quick. All right. Okay. So thank you so much for being here today, and it's a real honor to be selected for this wonderful seminar series. And I'm very excited to share with you the work that I've been doing so far in my postdoc looking at primordial germ cells. So as Wade mentioned, in my postdoc lab, the McDowell lab, we use adaptive light sheet microscopy. And we do this to understand these really historically inaccessible stages of mouse development that occur after the embryo has implanted into the uterus. And so this technology has enabled us to see a ton of different things, but my specific focus is primordial germ cells, which in this movie are shown in this sort of reddish pinkish color. And so primordial germ cells are really important cells. They eventually give rise to sperm and egg. And so they're obviously very important cells. And this microscopy is enabling us to look at the journey that these cells take in the embryo, in intact embryos, in a way we've never been able to before. So I'm going to give you a bit of background on primordial germ cells, or PGCs, as I'll call them for the rest of the talk. So PGCs are specified from the cells of the embryo after implantation at about embryonic day, or E6.5. And so after this, they begin this lengthy and complicated migration through the developing tissues of the embryo. And this journey takes multiple days of development. So from E7.5 to the first PGCs arriving at their destination at around E9.5. Once they've arrived at their destination, which is the site of the future gonads, the gonads will form and the PGCs will mature, giving us sperm and egg. And then upon fertilization, the cycle continues. And so I like to argue that PGCs are the most important cells in the embryo because they are the only cells that go on to produce future generations. So not only are PGCs important developmentally, they're also a fascinating model, in my opinion, for 3D cell migration in vivo, because they naturally migrate through diverse environments. And so we know from a lot of cell culture work that if you put cells in different types of engineered environments, they'll exhibit different types of migration modes, like protrusive or amoeboid. And so PGCs are a really cool system because they naturally move through many different tissue types. So at E7.5, they go from the epiblast at the middle of the embryo through the mesoderm and then out to the endoderm that's covering the surface of the embryo. At E8.5, they remain in that endoderm, but that endoderm starts undergoing pretty serious morphogenesis. So it goes from being a surface layer on the edge of the embryo, and it turns into a tube that goes inside the embryo to form the hindgut. And so that morphogenesis is obviously associated with a lot of changes in the shapes of cells in the hindgut, which may have an effect on how PGCs are moving through it. Finally, when we get to E9.5, PGCs will leave that hindgut epithelium or endoderm and migrate through the surrounding hindgut mesentery to get to their final destination, which is the gonadal ridge. And so one of the questions that I had when I started learning about this system is trying to understand exactly how PGCs are adapting to these different environments. And especially, can we sort of compare what we find in the mouse to what has been done in cell culture to understand how these different migration modes that we've observed in that context might be deployed in vivo. So when we think about migration mode or how cells are moving, one of the simplest readouts of that is to just look at the shapes of the cells. So that was the starting point. And so here we're using a variety of different labeling strategies to look at cells. So at E7.5 in the early stages, we can use a PGC-specific Cree to highlight their cell membranes, and then a mosaic Cree to highlight membranes of somatic cells so that we can see all the fine protrusions that they're making. And at these stages of development, both PGCs and somatic cells are extending these long, thin protrusions between themselves. And overall, the cell bodies of both somatic cells and PGCs are the same area and have the same overall shape, quantified here as roundness. If we look slightly later, though, once PGCs are arriving at the endoderm, and while they remain in the endoderm from E7.5 all the way to E9.5, they tend to exhibit these much rounder shapes that have a greater cross-sectional area. So this may suggest that the PGCs are incorporated into that endoderm layer slightly differently, and or that they may have different mechanical properties that cause them to remain rounded while deforming their neighbors. Finally, when we get to E9.5 and they're leaving the endoderm and moving through the mesentery, we start to see some really amazing protrusions. So here we're again just using that mosaic label to show somatic cells, individual somatic cells, and then a PGC-specific antibody that really nicely highlights the membrane here. And so we've, at these stages, the protrusions are much longer than those of somatic cells, and yet they still maintain that more rounded cell body. And so we did a little bit of Aries scan imaging to get a better idea of what these protrusions look like. And what we found is that even on a single PGC that's in the mesentery, we can see many different kinds of protrusions, from these big wide blebs down here to these really thin filopodial protrusions coming out at this end. And then, of course, these cytoplasm-filled ones that we saw here as well. So we're interested in understanding whether these protrusions are part of how they're migrating, because different protrusion types are associated with different migration modes. So more thin protrusions, possibly cytoplasmic-filled protrusions, are associated with a more mesenchymal type of migration, whereas blebs are associated with a more amoeboid type of migration. So to understand this, we turn back to some live imaging. So in this example that we're looking at here, once the slide changes, there we go. We have PGCs labeled and all cell nuclei are labeled. And so we can see that as PGCs are moving around in the hindgut endoderm here, that they are extending these protrusions that we can see well enough with a cytoplasmic marker. If we carry out a similar experiment using mice that express a PGC marker, but also an actin reporter, we can see that these protrusions tend to be actin-rich. So if you look for the little flashes of white protruding out from the cell, you can see that that is the co-localization of our PGC marker and the life-act RFP. And so these protrusions are actin-rich. And this was really surprising because one of the other model organisms where we know a lot more about PGC migration than we do in the mouse is zebrafish. And in zebrafish, PGCs use a form of blood-based amoeboid migration where that's characterized by this sort of bleb at the cell front shown here that is completely devoid of actin. And so we're finding that PGCs in the mouse may be operating using a completely different migration mode than what is seen in zebrafish. So the other important determinant of how cells are migrating is their surrounding environment, as I keep saying. And so the next thing we wanted to look at was how the environment around PGCs changes over the course of their journey. And by environment here, I'm today just going to talk mostly about ECM. So if we look at E7.5 at the beginning of this migration, when PGCs are moving out to the edge of the embryo, they're moving through the mesoderm. And if we stain for different ECM components, we find that there's this sort of highway of ECM that's enriched along the path that PGCs are taking through that tissue. when we perform live imaging of a mouse that expresses both a PGC marker and a reporter of laminin, we find that the arrival of PGCs in this part of the embryo, in this mesoderm area, coincides with the appearance of laminin. And so this is suggesting that the development of this ECM pattern is closely linked to the migration of PGCs themselves. Looking slightly later at development, we see that PGCs continue to have this really close association with laminin. So once again, using this laminin B1 reporter shown on the previous slide, we looked at hindgut explants and noticed that the PGCs seem to have this accumulation of laminin all around their edges. So much so that you can almost pick out PGCs without even having the PGC marker present. So we confirmed this with two different laminin reporter lines, B1 here and C1 here. And in both cases, PGCs are surrounded by laminin when they're in the mesentery and when they're in the hindgut endoderm, which is really surprising because you don't normally think of there being a lot of ECM inside of an epithelium like the hindgut endoderm. And we confirmed that PGCs are indeed expressing laminin using HCR in situs. But these observations raise this really interesting possibility that perhaps PGCs are sort of constructing their own ECM environment around themselves as they're migrating. And so we're trying to think of interesting ways to follow up on that idea in future experiments. So it seems that ECM may serve as somewhat of a scaffold for PGCs as they're undergoing migration. But ECM can also, instead of facilitating migration, it can also hinder migration. And this is sort of what we think of as the role of the basement membrane. So the basement membrane that PGCs have to encounter is the one that is underneath the endoderm and then eventually surrounding the hindgut epithelium. So if we look at the formation of this basement membrane over time, we can see that at E7.5, if we look at different ECM components, that they're present at the basal surface of the endoderm, but kind of in a really patchy way. And then as a development proceeds, as we go from E7.5 to E9.5, that basement membrane increases in thickness and also improves in terms of continuity. So the basement membrane is really being elaborated and developed over the course of this journey. And so if we think about those changes in the basement membrane and what events are happening in PGC migration, we can sort of formulate some interesting hypotheses. So at E7.5, for example, when there isn't much of a basement membrane, this might mean that there isn't much of a barrier for PGC entry into the endoderm. Conversely, at E9.5, when the basement membrane is quite robust and really present, this might serve as a barrier to PGC exit, which could help keep PGCs in the endoderm for hindgut morphogenesis, but then also produces quite a dilemma for PGCs because then they're going to have to break through that basement membrane when they exit into the mesentery. So we wanted to look at these sort of transition events a little bit closer. If we look simply at immunostaining, we can look at how the shapes of PGCs as they're entering the endoderm and exiting the endoderm. And we find that at E7.5, before you have a robust basement membrane in place, that PGCs retain very nice rounded shapes when they're entering the endoderm, suggesting that it's kind of a relaxing process for them. Whereas at E9.5, we see much more significant cell deformations, suggesting that the basement membrane is indeed creating a barrier that they need to squeeze through. We corroborated this with some live imaging as well. So in this light sheet movie, which is coming, there it is. In this light sheet movie, we can watch, oh, there we go. In this light sheet movie, we can watch PGCs as they are sort of arriving at the endoderm on the surface of the embryo. And what we notice and what's sort of shown here in these stills is that PGCs can arrive at the endoderm and leave the endoderm. And if we quantify the distance between individual PGCs and the surface of the embryo, we find that these sort of fluctuate. And so this sort of transient entry, exit, re-entry is common to PGCs at these stages, consistent with the idea that the basement membrane is not providing a significant barrier and that it's an easy thing for them to incorporate into the endoderm. To start thinking about the exit process, which we're hypothesizing is a little more difficult, we looked closely for PGCs that were leaving the hindgut endoderm and moving into the mesentery. And we observed some really interesting things. And this was one of the earliest images I took in my postdoc. And Kate and I were both just like, what is going on here? Because what we have here is a PGC where the entire cell body remains in the hindgut endoderm, but these long, thin protrusions are extending into the mesentery. We perform live imaging, of course, to better study these protrusions. And in this lovely movie here, where we have a PGC marker and we also have laminin to show where the basement membrane is, we can see that the protrusions are indeed extending through the basement membrane and that they are extremely dynamic. And so I've included stills here just in case. And these are just some example traces of protrusion length over time that show how dynamic they are, including an example of a cell here that has 3 different protrusions that it extends and retracts into the surrounding mesentery. And so these sort of dynamic and long-range protrusions might be searching for signals that will promote migration or exit from the endoderm. And another thing that they could be doing is starting to clear a path through that basement membrane so that PGCs can exit the hindgut endoderm. So we followed this process a little further. And so in this movie, I want you to focus on this cell marked here in the middle and watch as it exits from the hindgut endoderm to the mesentery. And so what you'll see is that that long thin protrusion appears as we saw before, but gradually it widens and then the entire PGC exits from the endoderm. And so we can sort of quantify this characteristic behavior or this sort of characteristic sequence of events that characterizes exit. So initially we have a protrusion. At a certain point, it becomes much wider, and this is followed by elongation of the cell body itself and exit of the PGC from the endoderm. And so this protrusive migration that we're seeing as they're exiting is something that continues as they're migrating through the mesentery. So we see sort of 2 different migration types when we're looking at PGCs moving through the mesentery. So the first is a really nice protrusive migration, like here, like I show in this movie here. So in this example, you have a cell with no protrusion, then it forms a nice long protrusion, the cell body elongates, we have a nice alignment between the protrusion itself and the cell body, and the cell moves. And so this is very orderly, beautiful process. The other behavior that we see a lot is this sort of non-migratory or so this other type of migration where the cell is still extending protrusions in many different directions, but it's not really going anywhere. And so these 2 different behaviors, we were noticing that they happen to be correlated with sort of differences in the surrounding tissue. And so you may have already noticed just by looking at these movies that the first movie that I showed where the PGC is undergoing this really nice directed migration, we have, come on, next slide. We have lower cell density. There we go. Yeah, so lower cell density and this like sort of faster directed migration. Whereas in regions of higher surrounding cell density, we have this slower, more meandering migration. So PGCs are potentially sensing the density of surrounding cells and looking for the right path to follow so that their migration is easiest. So this got us into thinking about intercellular spaces in the embryo and in the endoderm. So a simple dextran experiment to highlight these intercellular spaces showed that the intercellular spaces are around the same size as PGC protrusions, which makes sense if PGCs are extending protrusions and figuring out where to go, but they are significantly smaller than the PGC cell bodies and the PGC nuclei. And so in order to get through these intercellular spaces, PGCs need to deform pretty significantly. And so in this, I have too many movies, and so everything's slowing down. In the movie that will appear shortly, there it is, we can see based on the cytoplasmic marker that the cell body deforms pretty significantly during migration, and based on the nuclear marker that the nucleus is deforming as well. And so basically what we call this is that PGCs are migrating under confinement, which is an important area and kind of a hot topic in certain cell culture fields. Because what we've learned recently from experiments done in which cells are forced to migrate through narrow microfluidic channels, that sort of confinement is sufficient to cause rupture of the cell nucleus and DNA damage, which can lead to significant issues for the cell and for the nucleus. And so in PGCs, we started to try to think in that framework and understand what are the negative consequences of this confined migration for PGCs. So in this example, we see a PGC that is exiting from the hindgut endoderm, and this is a completely well-typed situation here. As soon as it exits, it fragments into little pieces and dies very quickly, suggesting that that exit through the basement membrane may have had really significant consequences for the cell, causing it to rupture. We can also explore the role of confinement by tuning confinement, and we did this by treating hindgut explants with a matrix metalloprotease inhibitor, or MMP inhibitor. And so in the control, you can see PGCs are nicely extending protrusions and moving around. But in the treated situation, we see something quite different. So I want you to follow the cell that's marked here. When the movie starts, you'll see that this cell is still making protrusions and it's still moving, but it squeezes and deforms really dramatically as it moves through this space and then immediately ruptures into little pieces. And so what I've quantified down here is these fragmentation events. And so increasing the amount of confinement by inhibiting MMPs leads to increased fragmentation of PGCs. And in certain cases, we've even observed PGCs that have a ruptured nucleus. And so this is an example that I found with one of my high school students last summer of a PGC that had its nucleus broken into 2 separate pieces. So there are significant things that can happen to PGCs as they're migrating in confinement, and we don't even need to include this, this treatment for these things to happen. Both of these things happened in totally untreated conditions. So as I mentioned, the microfluidic cell culture work showed that these confined migration events can lead to DNA damage. So we looked at that next. If we look at the early stages of development, so before PGCs have migrated very far, we find that DNA damage is low in all cells, so PGCs compared to the surrounding somatic cells. But if we compare this to E9.5 after PGCs have, navigated basement membranes, endoderms, and mesentery, they seem to have a greater accumulation of DNA damage markers than their surrounding somatic cells, consistent with this hypothesis that confined migration can lead to DNA damage in these cells. So this is really surprising because PGCs are extremely important cells, as I have been arguing since the beginning. And so why would evolution have led to this really risky confined migration process for these super important cells? And it's possible that it has also conferred some advantages or some adaptations to these cells that will enable them to cope with the effects of confined migration. And one of the ways they could do this is through changes to the mechanics of their nuclei. And so in line with this, we find that over the course of development, PGCs deplete lamin B1 in the nuclear envelope. So I'm just showing you E7.5, so before migration, and then E9.5 towards the end of migration, but every stage is quantified here. We have this really significant depletion of lamin B1, which could lead to softening of the nuclear lamina. At the same time, PGCs are upregulating lamin B2, which can also be associated with softening of the nuclear lamina. And so it could be that they're completely changing the mechanical properties of their nuclear lamina and making them softer, which may allow them to squeeze through spaces unharmed. And so in line with this idea that the nuclear membrane is softer, we stained embryos with a nuclear pore complex protein to show the shape of the nuclear envelope. And throughout their entire development, PGCs have a way more deformed nuclear membrane. And so we see a lot of these little invagination points suggestive that it's much softer and it's producing this really wrinkled morphology of the nucleus. And so we're interested in understanding whether these changes to the nuclear lamina may help PGCs to protect their nucleus and their DNA integrity during the confined migration that evolution has, for some reason, forced them to undergo. So I just want to sum up. So in closing, I've shown you today that PGCs undergo a remarkable migration through diverse environments of the developing mouse embryo. And they use migration modes that seem to be distinct from those in other well-characterized model organisms. They remain closely associated with ECM throughout their journey, which provides a possible scaffold or guidance cue for their migration. And then at the later stages of migration, significant confinement is imposed on PGCs, which necessitates cell and nuclear deformation, and sometimes causing DNA damage and cell death. And so overall, the picture that's sort of starting to come together here is that the development of the embryo and the tissues and basement membranes around PGCs causes increasing confinement and physical stresses on PGCs throughout their migration, potentially requiring PGCs to adapt at the cell and nucleus levels to ensure that they successfully get to their final destination. And just finally, I hope I've convinced you that this is a really exciting and fascinating model of in vivo cell migration. And I hope to build a lab to study this process one day. So if you think this is an exciting model and it would be a good fit in your department, I would absolutely love to talk. So in closing, I would just like to thank my supervisor, Kate McDowell, who refuses to be in pictures, so she's behind this plant here. My wonderful lab mates, the Crossan lab at the LMB, who've been helping me with some of the DNA damage stuff, our light microscopy facilities, and of course our mouse house. And this is a triplet embryo that I found last year that I'm very proud of. So with that, I'm happy to take any questions. Thank you.
Well, thank you, Catherine, for that beautiful talk. The floor is open for questions, so people can just type those into the Q&A. I'll read the first one that's already there. Catherine, you might already be able to see it. It's a little long. This question comes from Grant McGregor. Thanks for a beautiful talk. From static images, mouse PGCs have been described as migrating like an elephant walk, as in where elephants walk in a line with a lagging elephant holding the tail of a leading elephant. In vivo, is this really what happens? Do PGCs extend filipodial cytonema-like projection in the direction of migration while also extending some sort of filipodial structure at its trailing edge? Or is there no tail? Do you ever observe filipodial cytonemal structures projecting from one PGC beyond the nearest PGC? And can you speculate on the nature of diffusible versus non-diffusible factors that direct the leading PGC towards the gonadal ridge and the potential of filipodium cytoneme in this process? Do you think the leading PGC has a different identity compared to the lagging PGCs? This will answer many people's questions at once. So,
okay, good. Yeah. I have the question open as well, so I won't make you repeat any of it. But thank you. Who is this? Thank you, Grant, for the wonderful questions. Yeah, so I have not seen this sort of leader follower or elephant walk. I hadn't heard that term before. I haven't really seen anything like that. They do really seem to be behaving on their own while they're moving through the mesentery. Sometimes they end up in little groups, but they aren't necessarily moving when they're in those groups. They sort of hang out together, form protrusions to each other, and then go on their way. And so the leader follower dynamics, it may be there, but if it is there, it's not immediately obvious to just like the viewer. The filipodial and cytonemal structures are absolutely fascinating. I would love to talk offline about what these are doing and what sort of signals are they receiving. So when they're first extending, like one of the main questions in PGCs is how do they know when it's time to exit the endoderm? You know, that's an excellent question. We have no idea. But these little protrusions that they make right before they exit may have something to do with receiving or looking for that signal that tells them it's time to go. So I don't have any great answers, so I'll move on from that. But if you ever want to talk offline, I would love to.
Thank you very much. The next question is from Emilio Mendez. Nice talk. Do these protrusions have primary cilia? Thanks.
Great question. I have not looked for primary cilia at all. I haven't thought of that. I guess every cell has a primary cilium, and PGCs are kind of like embryonic cells or like cells of the early embryo. So yeah, I haven't looked at that. Very good question.
The next question comes from Andrew Lopez. Really great talk. I wanted to ask if you could expand on why PGCs seem to go through a gauntlet. It's really interesting. They have to soften their nucleus to survive. Do you believe that there is a greater purpose?
Amazing question. Again, I could talk about this forever because it is really counterintuitive. There are a few sort of possibilities. So one possibility is that they go through the gauntlet for a survival of the fittest type thing. So maybe only the PGCs that are most able to navigate the embryo and maintain the integrity of their genome are the ones that get to be at the gonads and therefore produce future generations. Another possibility is that any mutations that are generated by this DNA damage generate diversity. And so that could be a way for organisms to have diversity in their offspring. I have no evidence for any of these things, but it's a really fun thing to think about. And I hope in my career to design experiments to get at some of these things. So thanks.
The next question is from Bridget Wass. Really interesting work and beautiful images. Is it known whether these protrusions are detecting certain morphogens / attracting cues?
Yeah, so there are certain chemokines that are expressed. So there's one, for example, that's expressed by the gonadal ridges that is thought to sort of direct PGC migration through the mesentery. Whether those are those are sort of received by receptors on the protrusions themselves, I don't know at all, but that would be really interesting to look at.
The next question is from Ankita Tawani. Can you talk about if the cell guidance molecules are different in E7.5 versus E9.5 when the field of cells to navigate are looser versus more restrictive, respectively? Great talk, lovely movies.
Thanks. So unfortunately, we don't know a ton about the guidance signals in this process. We know about certain ligand receptor pairs that are expressed by PGCs and the surrounding cells on their migratory path that may provide a survival signal or a migration cue. And we know of some diffusible molecules that they migrate towards. I do think that they are different at E7.5 and E9.5 if we're talking about diffusible things, but that sort of survival signal I mentioned of the ligand receptor pair, that is present, I believe, throughout the migration. So we really don't know much about it, but I think it would be great to study this further with, for example, spatial transcriptomics or something, so we can see what possible signals are around that they're moving towards.
Thank you. So the next question is from Seven Hopian. Beautiful, thank you. Do you think the roundness of some of those cells suggests they lack adhesion affinity with their neighborhood and are therefore motivated to move on?
Great, thanks for the question, Sivan, and hello. That's an interesting question, yeah. So at the early stages, so when they're moving through the mesoderm, like all cells have a little bit of ECAD on them at that point. And so there is a possibility for some sort of cell-to-cell adhesion there. Once they're in the endoderm, everyone has ECAD as well. And then very strangely, when PGCs leave the endoderm and move into the ECAD negative mesentery, they retain ECAD at their membrane. So there's definitely something strange going on with cell adhesion. Whether it's sort of like an adhesion-based sorting, I don't know if that's entirely consistent with what we're seeing. So if they were being sorted out of the endoderm, for example, when they're going into the mesentery, I don't think we would see these little protrusions coming out first. I think we would see the cell bodies coming out first as they're being forced out due to differences in adhesion. But it's definitely an important question, and yeah, it would be great to study the various cadherins that are at play here.
I had a question for you. Can you comment more about the differences between this mouse system and zebrafish? Because your ability to do live imaging there, of course, really cracks one of the hardest things about this cell type and that event is just highly dynamic. So I wanted to know if some of the difference between the scale of it, between how far these cells have to go in a mouse versus how far they have to go in a zebrafish, how long it takes, does that impact at all on why you're observing differences in some of these molecular mechanisms? It's more a function of different timing rather than, if that makes sense.
No, it does. It's a great question. I haven't given it much thought. I think the migratory path is kind of different between mouse and zebrafish. It's actually more similar between mouse and flies than it is between those and zebrafish. If I'm not entirely mistaken, I think the zebrafish ones spend more time in an endoderm that's a little bit more like the endoderm at E7.5 in the mouse. And so it could be that the sort of rounded morphologies that I'm seeing at that stage are a little more consistent with what is seen in zebrafish. But then as the journey, extends over several days in the mouse and tissues are really being developed, by the time they leave the hindgut, that's an epithelium. It's not just an endodermal layer. And so that might necessitate differences in how these cells are migrating through them. It could be that amoeboid isn't sufficient for them to move between these cells, and they really need protrusions to clear the way. Great question, and that's something I'll be thinking a lot about.
All right, thank you very much. So we got time. If there's anybody who's real desperate to, here we go. You do have a question from Celeste Berg. Are any of the signal receptor pairs conserved in flies?
I believe they are, but I confess that I don't know as much about flies as I should, but I think that there is some conservation between these things. But of course, we know a lot more about PGC migration in flies than we do in mouse, so it's possible that things that are happening in flies, just we don't know in mouse yet, because mouse is so unwieldy compared to the beautiful Drosophila embryo. So
I think we have time for one last question, again from Grant McGregor. Have you ever observed PGCs that become lost during migration? If so, do they still have a philopodium cytoneme? And if so, in which direction does it point, i.e. towards the gonadal ridge?
That's a great question. So yeah, PGCs do become lost along the journey. Typically they undergo apoptosis, but it's thought that they can stick around and give rise to germ cell tumors. So I have found a couple of PGCs that were definitely in the wrong place as late as E9.5. They don't seem to have quite the same protrusive activity, but this is a hard cell to study because they can really end up anywhere. At the early stages, they get swept off into the extraembryonic region. So it's a really tricky thing to study, and my N is like literally 2. So I can't answer your question with any confidence, but very interesting question.
All right, thanks, Katharine. That's almost the hour, so we'll go ahead and stop there. I want to thank both Ekasit and Katharine for their great talks. This seminar has been recorded and will be available on the SDB website next week. Please join us next month for the final seminar in this current series on Friday, May 10th, where Ankita Tawani from Baylor College of Medicine and Konstantinos Kaliviadis from Imperial College London will present. Thank you all for coming and enjoy the rest of your day.